Merci beaucoup, Amélie. On passe maintenant à Tasnim Mou.
Hello everyone, I am Tasnim. Today I am going to present about winter cyanobacterial bloom. What comes to your mind when you first hear about cyanobacteria? Definitely some harmful poison that could prevent your holiday fun in lake water. Well, this cyanobacteria is also important, you cannot imagine. In the beginning of the earth, cyanobacteria is first organism that started photosynthesis and were responsible for amount of oxygen that we breathe today. Under the preferable conditions, cyanobacteria may start intense proliferation and accumulation and form cyanobacterial bloom. Sometimes it causes the ecological degradation, which can be life-threatening because it produces toxin into the water. This toxicity is associated with cyanobacteria contaminated food, drinking water, and other recreational activities. Cyanotoxin may induce several health hazards and even death in human body. Cyanobacteria growth and development are highly influenced by several factors like temperature, nutrient, light, and water movement. Recent observation in Abitibi region shows some unusual occurrence of cyanobacteria plantothix under the ice cover, which contradicts the previous study that cyanobacteria always need high temperature for their growth. So our objective is characterize the cyanobacteria community composition and measure the toxin concentration in all over the year, even all climate conditions, and also determine the driving environmental and biological factor that influence the cyanobacterial bloom formation all over the year. So our first hypothesis is that cell activity, toxin concentration, and abundance of cyanobacteria will be detected all over the year, even all climate conditions. But even in winter in Lake Fortune, but only in summer in Lake Beauchamp. And our second hypothesis is that surface nutrient availability allows cyanobacteria bloom in Lake Fortune, and higher summer temperature allows cyanobacteria bloom in Lake Beauchamp. So for this, we conducting our study in two different lake in Abitibi Tamskiming. One is Lake Fortune and another is Lake Beauchamp. We collect water sample from 3 stations and 3 different layers of the lake to measure some physiochemical variation and also measure some temperature, pH, conductivity, zooplankton and phytoplankton community. RNA analysis will tell us the cell activity, and finally we will identify the species that is causing the cyanobacterial growth in all over the year. So our preliminary results suggested that the dominance of cyanobacteria, plantothix and limnothix during the July to September. We found higher nutrient concentration from July to September. So the outcome of this project is to contribute the goal of life-threatening toxicity producing cyanobacteria all over the year. Thanks for the attention.